Introduction of CowellBMP. Factors of bone regeneration. What is BMP2? Why to use BMP2? Why to use rhBMP2? History of CowellBMP. How to do recombinant process. Comparison with Infuse. Manual of CowellBMP. In large defect of extraction circuit, the natural healing potential could not be complete. Even in 1 year and 1 month after extraction, the extraction circuit was not healed. The tissue engineering technique has to be applied in the large defect. The vertical height could be decreased even in the graft circuit. As a result, the deep pocket around implant could be developed and the oral hygiene could be difficult from the high gingival level. GBR technique could prevent the loss of vertical bone height and width in the limit scale. The basal bone of maxilla and mandible could be severely lost for long period after tooth missing. The dimension of ridge has to be augmented for implant placement. The autogenous bone graft is recommended with GBR technique. The disadvantage of autogenous bone uptake is severe bleeding, swelling, post-operative pain, and complications. The failure of autogenous block bone could be occur in unignorable frequency. The growth factor like BMP2 is necessary with autogenous bone and the others. Researchers tried to promote the healing and regeneration of tissue. The key factors of tissue regeneration are scaffold, stem cell, and signal molecular. The scaffolds support the volume and shape of the regenerated tissue, as well as osteoconduction growth of cells. This is composed of the organic and inorganic particles or block type. The stem cells supply the proliferation of the regenerated tissue with osteogeneration property. This is originate from the living tissue of patient. The signal molecular stimulates the stem cells differentiation and proliferation as well as osteoinduction of cells. This is supplied by the recombinant gene technique. PCL-graded synthetic bone is grafted with rhBMP2 in the mandibular defect of dog. In post-operative X-ray, PCL bone material is radiolucent. On 8 weeks after graft surgery, the defect grafted with BMP2 shows a radiopaque appearance in whole area due to the new bone growth. But the defect grafted without BMP2 shows a radiopaque appearance only in bottom. In conclusion, BMP2 stimulates and proliferates the osteoblast of the surrounding bone around the defect. Autogenous bone is grafted with rhBMP2 on the perforated defectum fixture. On 3 weeks after graft surgery, the defect grafted with BMP2 shows that the new intact cortical bone was regenerated and remodeled with the remained bone. BMP2 stimulates and proliferates the osteoblast of the surrounding bone around defect and the grafted autogenous bone. In conclusion, the best graft material is the combination of autogenous bone and rhBMP2. BMP2 is a group of polypeptides. This function is involved in cellular proliferation and differentiation and morphogenesis of tissue and organ during embryogenesis, postnatal growth, and adulthood. BMP2 is a non-collagenous protein not broken down by colinase during infection and is dimeric protein that is composed of two subunits linked by disulfide bonds. BMP2 acts on the stem cells. The cellular mechanism is below. BMP2 binds BMP receptors in stem cell membrane. BMP receptors is two kinds composed of integrated form and the separated form with BMP receptor 1 and 2. The separated BMP receptor 1 and 2 bound with BMP2 activate cell proliferation and differentiation. The integrated BMP receptor 1 and 2 bound with BMP2 activates intracellular phosphorylating enzymes, then induces intracellular signaling pathway of SMAD. SMAD migrates to nucleus and activates set of VEGF, BMP, and Twist2 genes. As a result, specific change in cellular activity or phenotype is occurred. A maturing stage of cellular proliferation and differentiation, Twist2 growth factor regulates the overgrowth. Why to use BMP2 in implantology? Titanium implants were placed into calcium phosphate block. This block was saturated with rhBMP2 and implanted into subcutaneous tissue of rat. On 8 weeks after implantation, the new bone was induced into block and contacted with implant surface. But the block without rhBMP2 saturation was filled with fibrotic tissue. In conclusion, the immediate implant placement and simultaneous bone graft in defect and extraction socket could be successful. The grafted site will be filled with the new bone and the implant surface of grafted site will be osseointegrated. How much the effective dose of BMP2? rhBMP2 dose test was done in red calvarial defect. Amount of new bone was measured at 8 weeks after surgery. The effective dose was from 2.5 mg to 20 mg dose of rhBMP2. The response of bone regeneration was dose independent. Without rhBMP2 saturation, the cortical bone was not formed and the percentage of the new bone was significantly decreased. The minimal dose was 2.5 mg, which is lower tenfold than the maximal dose 20 mg. In clinical application, 0.1 gram of rhBMP2 was effective in the various volume of the defect and extraction socket. Why is recombinant human BMP2 need? BMP2 is existed about 2 nanogram per gram in native bone. Effective dose of BMP2 is about 0.1 mg per gram. 50 kilograms human bone is needed for 100 mg BMP. Mass production is impossible. For mass product, human BMP2 gene could be transplanted to the bacteria and the animal cell. The cell is cultured and secretes BMP. BMP is collected and purified for clinical uses. CowellBMP was developed from 2004 year. rhBMP2 was first purified from 2004 year in CowellMedi Tissue Engineering Research Center. Preclinical study, safety, Effectiveness, dose, and release time were researched from 2004 to 2007. Clinical studies have been done from 2008 to present in extraction socket, maxillary sinus, periodontal defect, and open membrane technique. Approval of KFDA was done in 2010. Total 20 research articles were published in SCI Journal. How to do recombinant process. The process is a serial sequence of the gene cloning of human BMP2, protein overexpression using E. coli system, purification of the rhBMP2 protein, and dimerization of the rhBMP2 protein. E. coli produces monomer protein of rhBMP2, which is inactive. After dimerization process of monomer protein of rhBMP2, rhBMP2 have the activity of itself. Comparison with infuse. Infuse is origin from Chinese hamster ovary cell. CowellBMP is origin from E. coli. Infuse scaffold is collagen sponge. CowellBMP is TCP-HA particles. Infuse scrap volume is severe decreased. CowellBMP's is stable. Infuse dose is a lot. CowellBMP's is maximal 1/10 fold than infuse, which is safe in adverse effect. Infused BMP solution is easy to escape from surgical site. CowellBMP's is difficult to escape because the solution is injected into the graft site after primary closure of wound. 0.1 gram CowellBMP is composed of 0.1 mg BMP powder and 0.1 gram TCP-HA ceramics. BMP powder is attached on bottom of bottle. TCP-HA powder is poured in sterilized dish. From 0.1 to 0.2 cc sterilized water or saline is injected. BMP2 powder is soluble with water. From 0.1 to 0.2 cc BMP2 solution is aspirated by 1 cc syringe. TCP-HA powder and the other bone graft is filled in the defect. The wound is closed with flaps. From 0.1 to 0.2 cc BMP2 solution is injected through flaps into the graft site.